Namaskar. Welcome to the session on proteins and enzymes. Proteins are defined as high molecular weight polymers of alpha amino acids that are formed by living organisms. The primary structure of proteins consists of a polypeptide chain of amino acids residues joined through peptide bonds. Milk contains a heterogeneous mixture of proteins. Normal cow and buffalo milk contain about 3.5% proteins. The concentration changes significantly during lactation, especially during first few days of postpartum. The greatest change occurs in the whey protein fraction. The natural function of the milk protein is to supply the young mammals with the essential amino acids required for the development of muscular and other protein containing tissues, and with a number of biologically active proteins, that is immunoglobulin, vitamin binding, metal binding proteins and various hormones. The young of different species are born at different states of maturity and consequently have different nutritional and physiological requirements. These differences are reflected in the protein content in the milk of the species, which ranges from less than 1% to about 24%. The protein content in milk is directly related to the growth rate of the young one of that species. In addition, the properties of many dairy products depend on the properties of milk proteins, since the proteins, particularly the casein, contributes significantly to the body and texture of the products, although the fat, lactose and especially the salts exert significant modifying influences. In the present study, the proteins and enzymes of milk are covered under the following sections. First is the proteins, general structure, amphoteric nature, difference between casein and serum protein, different types of casein, that is acid and rennet, uses of casein, fractionation of protein. Second is enzymes, catalase, alkaline phosphatase, lipases and proteases. Classification of milk proteins. Table 1, broad classification of milk proteins. First column is the type of protein and second column is its concentration in milk. Alpha casein 15 to 19 gram per liter, beta casein 9 to 11 gram per liter, K casein 2 to 4 gram per liter. Whey proteins, that is lactoglobulins and immunoglobulins 5 to 7 gram per liter. IgG immunoglobulins 0.6 to 1.7 gram per liter. IgM immunoglobulins 0.5 to 1.8 gram per liter. IgA immunoglobulins, that is lactalbumins 2 to 4 gram per liter. Serum albumin 0.2 to 0.4 gram per liter. When milk is acidified to pH 4.6, the isoelectric point at room temperature, about 80% of the total protein in cow and buffalo milk precipitates out of solution. This fraction is called casein, and the protein which remains in the soluble in the solution is referred as whey or serum protein. The solution also contains small amounts of non-protein nitrogenous compounds also. The ratio of casein, whey protein, varies between the species. In human milk it is 40s to 60, while in cow, buffalo, sheep and goat milk it is 80s to 20. Casein in fresh raw milk is present in micellar form, mostly 0.02 to 0.30 micrometer in diameter, comprised of some 20 to 1500,000 casein molecules. These micelles also contain inorganic matter, mainly calcium phosphate, about 8 gram per 100 gram of casein. The casein micelles are voluminous and hold more water than individual casein molecule. The casein can be fractionated into alpha S1 casein, alpha S2 casein, beta casein and K casein. The caseins, especially alpha S2 casein, are rich in lysine, an essential amino acid in which many plant proteins are deficient. Hence, casein and skim milk powder are very good nutritional supplements for cereal proteins which are deficient in lysine. But due to high lysine content, casein and products containing casein undergoes extensive non-enzymatic Maillard browning on heating in presence of reducing sugars, that is lactose. The existence and properties of casein micelles have several consequences in the use of milk. The stability of milk products during heating, concentrating and holding is largely determined by these micelles. The rheological properties of sour milk and concentrated milk are largely determined by the changes occurring in these micelles. The interaction of these micelles with air-water and oil-water interfaces is important in homogenization. The casein micelles also influence some of the properties of the homogenized products. Amphoteric property. In chemistry, an amphoteric compound is a molecule or ion that can react both as an acid as well as a base. The amphoteric property of proteins is due to the presence of free carboxylic and free amino groups at the end of protein molecule. All amino acids contain at least two ionizable groups, the alpha amino groups and alpha carboxylic group, which are responsible for the acid-base behavior. Because of the amphoteric nature, the proteins act sometimes as acids and sometimes as bases depending upon the pH of their medium. Whey proteins. Whey proteins constitute about 20% of the total protein of cow and buffalo milk. Sometimes this fraction of protein is also referred as serum proteins or non-casein nitrogen. It has two well-defined groups of proteins called lactoglobulin and lactoalbumin. Lactoglobulin fraction consists mainly of immunoglobulins, especially IgG, with lesser amounts of IgG2, IgA and IgM. The lactoalbumin fractions contain three main proteins, that is beta lactoglobulin, alpha lactoglobulin and blood serum albumin. Major differences between the caseins and whey proteins. In contrast to the caseins, whey proteins do not precipitate from the solution when the pH of the milk is adjusted to pH 4.6. This difference in the properties of the two milk proteins is exploited in the preparation of industrial casein and varieties of cheese, that is cottage cheese, quark and cream cheese. Chymosin and some other proteinases known as rennet produce a slight but very specific changes in casein, resulting in its coagulation in the presence of calcium ions. Whey proteins do not undergo such alterations. Though coagulation property of casein through the action of rennet is exploited in the manufacture of most cheese varieties and rennet casein. Casein is very stable in high temperatures. Milk may be heated at natural pH around 6.7 at 100 degrees Celsius for a long time without coagulation, and it withstands the milk sterilizing temperature. The whey proteins on the other hand are heat labile, being completely denatured by heating at 90 degrees Celsius for 10 minutes. Caseins are phosphoproteins containing on an average of 0.85% phosphorus, while the whey proteins does not contain phosphorus. The phosphate groups are responsible for many of the important characteristics of casein. It helps in binding calcium and hence making it nutritionally rich protein, particularly for the young ones. The phosphate is also responsible for very high heat stability of milk. Casein is low in sulfur content, about 0.8%, while the whey proteins are rich source of sulfur, about 1.7%. The whey proteins are molecularly dispersed in solution or have simple quaternary structures, whereas casein have a complicated quaternary structure and exist in milk as colloidal form. Casein is synthesized in mammary glands and is found nowhere in nature. Some of the whey proteins such as beta-lactoglobulin and alpha-lactoglobulin are also synthesized in the mammary gland, while others such as bovine serum albumin and immunoglobulins are derived from the blood. Fractionation of proteins. The two major milk proteins can be separated by acidifying the milk to isoelectric point, that is a pH of 4.6, where the casein gets precipitated, and the whey proteins will be in the fractionation of milk proteins as shown in figure 1 and fraction of casein into casein fractions as shown in figure 2. Fractionation of proteins. Skim milk. Cool the milk to 6 degrees Celsius. Acidify with dilute hydrochloric acid to pH 4.6. Holding for 30 minutes and then filter. Weigh solid mass. Add saturated magnesium sulfate. Press. Filter. Casein. Precipitate solution. Lactoglobulin, lactalbumin. Fractionation of casein. Dilute sodium caseinate solution. Ultrafiltration at 10 kilodalton cutoff membrane. Retentate, permeate and enriched caseinates. Beta caseinate enriched fraction. Freeze dry, ultrafiltration, permeate, retentate. Discard beta enriched caseinate, freeze dry. The fractionation of proteins, particularly into casein and whey proteins, is of economic importance. The whey proteins have many physiological functions such as providing immunity of minor components to the target area in the body. For example, lactoglobulin can bind retinol and protect from oxidation and transporting it through the stomach to small intestine. The whey proteins are also used in many pharmaceutical formulations. The casein fractions are used in pharmaceuticals. Production of caseins. There are two main methods of production of casein on an industrial scale. They are isoelectric precipitation method, that is acid casein, and enzymatic method, that is rennet coagulation. Two types of acid casein are being made commercially, that is lactic and hydrochloric caseins. Acid casein manufacture, lactic acid casein. For the manufacture of lactic acid casein, skim milk pH of 6.6 is first pasteurized at 72 degrees Celsius for 15 seconds. It is then cooled to setting temperature around 30 degrees Celsius and inoculated with lactic acid producing bacteria known as starters such as Lactococcus lactis, subspecies Cremoris, Streptococcus thermophilus at the rate of 0.1 to 0.2% of milk taken. The milk is incubated without agitation for a period of 14 to 16 hours. During this period, some of the lactose in the milk is converted to lactic acid by the starter bacteria and the pH is reduced to about 4.6, causing coagulation of the casein and the milk. After the pH of the milk has reached 4.6 to 4.7, the coagulum is broken and cooled, that is heated to a temperature of 50 to 55 degrees Celsius. After a brief period of residence in a cooking under acidic conditions, the casein gets precipitated. After cooking, the whey is drained and the curd particles are washed thoroughly with water, pressed to remove excess water. Finally, minced and dried in hot air oven or using fluidized bed dryer. The dried casein is then milled to get particles of desired particle size. Acid casein is insoluble in water, but soluble caseinate can be prepared by dispersing the casein in water by adjusting the pH to 6.5 to 7.0 with sodium hydroxide to produce sodium caseinate. Sometimes potassium hydroxide is used to obtain potassium caseinates. The caseinates are usually spray dried. Mineral acid casein. For the precipitation of mineral acid casein, pasteurized skim milk at a pH of 6.6 is mixed thoroughly with dilute 0.25 mole per liter acid at a temperature of about 30 degrees Celsius to a pH of approximately 4.6. In this case, because of the very vigorous agitation and the short mixing time, the casein is precipitated as very fine individual particles in a liquid serum, that is whey, unlike the gel or coagulum formed in lactic acid casein manufacture. The acidified milk mixture is then cooked and processed similar to as described for the production of lactic acid casein. Rennet casein manufacture. When rennet casein is made, the skim milk is not acidified. Hence, the pH remains at 6.6 throughout the manufacture process. Following pasteurization of the skim milk, it is cooled to a setting temperature of about 30 degrees Celsius and microbial rennet is added at the rate of 1.5 to 2 grams for every 100 liters of skim milk. The rennet added milk is kept undisturbed till a firm gel is formed. The renneting process completes in 40 minutes. The clotted milk is then cut into 1 square inch cubes using wire knives and cooked, and the casein processed in a manner similar to that described for lactic acid casein. Most of the rennet casein is used in the manufacture of cheese analogues. The rennet casein is generally prepared from good quality fresh skim milk and hence they are used in food and pharmaceuticals. Yield. The yield of commercial casein is about 3 kg per 100 kg of skim milk. Uses of casein products in foods. Casein is generally not consumed as a food on its own. Casein products are used mainly as ingredients in foods for modifying the physical properties of that food or to provide nutritional supplementation. As a consequence, they usually form a relatively minor proportion of the food. The function and various usage of casein in the different food products are listed below. As edible applications of casein products: bakery cheese products, coffee whiteners and creamers, confectionery cultured milk products, yogurt, etc., high fat powders, shortenings and spreads, ice cream and frozen desserts, infant foods, instant breakfast and beverages, meat products, nutritional food bars, pasta, pharmaceutical soups and gravies, sports drinks, whipped toppings. As protein supplements, the casein molecule's unique ability to form a gel or clot in the stomach makes it very efficient in nutrient supply. The clot provides sustained slow release of amino acids into the bloodstream, sometimes lasting for several hours. Casein is available as hydrolyzed casein, where it is hydrolyzed by a protease such as trypsin. Hydrolyzed forms are noted to taste bitter, and such supplements are often refused by infants and lab animals in favor of intact casein. As medical and dental uses, casein-derived compounds are used in tooth remineralization products to stabilize amorphous calcium phosphate, that is ACP, and release the ACP onto tooth surfaces where it can facilitate remineralization. As non-food uses of casein products: adhesive for wood, for example plywood, adhesive for foil laminates and paper, coatings for paper and cardboard, horticultural spreaders, joint cements in wallboard, leather tanning, paints, photoresist, stock foods, synthetic fibers and textile sizing. Enzymes in milk. Enzymes are organic catalysts elaborated by living cells that catalyze many of the reactions. Apart from their specificity in catalyzing the reaction, they are protein in general. Milk also contains several indigenous enzymes as constituent of the milk. Consequently, some of the enzymes will be entering the milk which has not been utilized during the biosynthesis of the milk. Similarly, some enzymes get incorporated directly as a measure to protect the constituents after the production of milk. As such, the enzymes naturally found in milk play a very significant role not only during the processing but also during their storage. As many as 60 indigenous enzymes have been detected in cow and buffalo milk. Many of the enzymes indigenous to milk are technologically important due to the following reasons. Deterioration. The most important commercially important enzyme proteinase, acid phosphatase and xanthine oxidase, for example enzyme lipase, or for preservation of milk quality, example sulfhydryl oxidase, superoxide dismutase. Thermal history of milk, for example are alkaline phosphatase, gamma glutamyl transpeptidase and lactoperoxidase. Indication of mastitis, N-acetyl beta D-glucose aminidase, acid phosphatase. The concentration of several enzymes increases due to mastitic infection. Antimicrobial activity, lysozyme, lactoperoxidase. Lipase. Lipase catalyzes the hydrolysis of milk fat, causing hydrolytic rancidity in milk and fat-rich milk products. Cow milk contains about 1 to 2 mg milk lipoprotein lipase per liter. Some lipase is in the cream fraction. However, the native enzymes is in the skim milk fraction where about 90% is bound to casein micelles. The lipolytic system in most milk becomes active only when the milk fat globule membrane is damaged by agitation, homogenization or temperature fluctuation. Significance of lipase enzyme. The effect of lipolysis is the rancid flavor which becomes detectable when the acid degree value exceeds MEQ per liter, milliliter equivalent per liter. Lipolysis may also produce varieties of other effects. One of the most important and noticeable is the lowering of surface tension. The liberated fatty acids, especially their salts, mono and diglycerides, are responsible for depressing the surface tension of milk. On the other hand, the lipolysis plays a positive role in ripening of cheese. The fatty acids, particularly the low chain fatty acids, as a result of lipolysis during ripening of cheese, are responsible for the development of desirable cheesy flavor in the ripened varieties of cheeses. In addition to the fatty acids, the esters and other compounds of fatty acid collectives contributes to the cheesy flavor. Milk alkaline phosphatase. Milk contains several phosphatases, the principal ones being acid and alkaline phosphatases. Among the two phosphatases, alkaline phosphatase is of technological importance. Alkaline phosphatase is concentrated in fat globule, hence cream contains more amount of this enzyme than in the milk. Milk alkaline phosphatase is used as the method of preference for determining whether the milk has been pasteurized adequately. Inactivation of alkaline phosphatase by pasteurization is an index of destruction of Mycobacterium tuberculosis. The inactivation of phosphatase enzyme by easy and simple chemical methods can be determined. Reactivation of alkaline phosphatase. The UHT treated and freshly pasteurized milk, that is pasteurized by high temperature short time method, shows phosphatase test negative, indicating adequate heat treatment given to milk during processing. When the milk is stored at 30 degrees Celsius for 6 hours, the pasteurized or UHT treated milk answers positive for phosphatase test. Reactivation of alkaline phosphatase is of considerable practical significance since regulatory tests for pasteurization assume the absence of phosphatase activity. The sulfhydryl group released by the whey proteins during heat treatment is responsible for reactivation of the enzyme during storage. Catalase. This enzyme catalyzes the decomposition of hydrogen peroxide to water and nascent oxygen. Catalase activity is higher in mastitis milk and colostrum than in normal milk. It increases with the multiplication of bacteria. Crystalline catalase has been prepared from several sources, but a small amount is present in milk. It is not fully purified. Milk catalase has a molecular weight of about 21 lakh and its isoelectric pH is approximately 5.5 and it contains heme iron. Catalase activities of 300 micromole per minute per liter have been reported for milk. Hydrogen peroxide is a very effective chemical sterile agent, though it causes some damage to the physicochemical properties and nutritional value of milk protein. Mainly damage is by oxidizing methionine. It is used as a milk preservative in the United States for the treatment of cheese milk, but not permitted in our country. Excess hydrogen peroxide present in milk can be reduced to water and oxygen by treating with exogenous catalase enzyme. This enzyme is used to detect the presence of hydrogen peroxide in milk, which is added as preservative in milk. Proteases. Milk contains two main proteinases, plasmin, that is alkaline milk proteinase, and cathepsin D, that is acid milk proteinase, and several other proteinases. In terms of technological significance, plasmin is the most important among indigenous proteinases. Plasmin accompany the casein micelles on the rennet coagulation of milk and are concentrated in cheese, wherein plasmin contributes to the primary proteolysis of casein in cheeses. The exogenous proteinases are of great importance in cheese making. The use of rennet, that is rennin or pepsin, is the principal application in proteinases in food processing. Rennet is next to amylases among industrial application of enzymes. Protein hydrolysates, which are obtained by partial hydrolysis of proteins, are used as food flavorings in soups and gravies and in dietetic foods. The functional properties of milk proteins may be improved by limited proteolysis under controlled conditions by using proteinases. Acid-soluble casein are suitable for beverages and other foods can be produced by partial hydrolysis of caseins. Controlled proteolysis improves the meltability of cheeses, but excess proteolysis causes bitterness in the product. Limited hydrolysis of whey proteins reduces its emulsifying capacity but increases its specific foam volume. On the other hand, the heat-stable proteinases enzymes produced by psychrotropic bacteria causes age gelation defect in the ultra-high temperature treated milk during storage. That is, though the milk is sterilized by UHT treatment, the heat-stable proteinases gels the milk proteins and milk will have gel-like structure which is not accepted by the consumers.